today I'll be talking about an American placental, the pocket gopher, and I promise I'm studying Australian mammals too, but the echidna portion of my study is still underway. So today I'm presenting data that I've analyzed here under the supervision of my advisor, Dr. Vera Weisbecker, at the University of Tasmania. So to introduce you to the western pocket gopher, here it is in action. It's a placental rodent found across a wide swath of the western United States. And it's a dietary generalist, and as a subterranean rodent, it has to be. The way that it forages is by digging long tunnels underground and basically eating whatever it finds. An adult gopher like this can dig up to 10 meters a day, moving 50 liters of soil in the process. Now, as any gardener knows, soil is incredibly heavy, so this is very energy intensive. So it represents a very intense selection pressure. As you can see, this individual is using its forelimbs to move the soil, and we actually see some species in this group begin to use their teeth to dig as well. So this makes for a very interesting study system for several reasons. First, they're one of the most morphologically diverse rodents we know of, and two, they're found across a wide diversity of soil, so there's a variation in how much selection pressure each of these species has. So, with that, my study group included 10 distinct species or subspecies of gophers. Now, this phylogeny is supported by both nuclear and mitochondrial DNA, so we feel very confident in it. And you can see that they split into two main subgenera. So the top group, subgenus Thymomes, is usually thought of as being more ancestral, and the subgenus Megascapus is thought to be more derived. So as we look at this in relation to body size, we see again that subgenus Thymomes tends to be small, and subgenus Megascapus tends to be large. And this picture gives you an idea of the range that we see in this genus, which is pretty big. Related to soil type, again, these are found across a wide variety of soils, which ranges from this kind of ideal Goldilocks soil that's really nice to move, all the way up to really hard clay soils, which tend to be dense and very hard to break apart. And sort of intermediate between those two ends of the spectrum is the sandy soil that's soft, so it's still easy to move, but tends to be more heavy. So this is a system that we're working with, and it allows us to create some hypotheses. But first, I hope that I've convinced you that digging is very intense selection pressure. And one of the most interesting adaptations that is driven is the development of tooth digging in some of these species. So tooth digging is very good in hard soils because, as you know, teeth are the hardest material in the vertebrate body, so it's harder than the keratin that just claw digging species can use. However, in order to use teeth consistently, they basically need to stick out, be buck-toothed. And we measure this as a degree of percumbency, so you've got to look like this backhoe here. And you can see that that angle of percumbency I pointed out with the red line is above 90 degrees. The constraint, however, for rodents is that their front teeth, their incisors, grow continuously in an arc. And so the ancestral condition has this angle of percumbency that's less than 90 degrees. So in order to dig with the teeth, we've got to get percumbent incisors. There's two ways to do so. The first is just to get bigger. And as a product of getting bigger, the radius of the incisor gets larger and it just sticks out more, so increasing that angle of percumbency. Second is a little bit more advanced, and that's to move the incisor root. And that is relative to the rostrum, so you get this more derived position here. And the way to distinguish between these two ways of getting more percumbent is to look at the rostrum. You'll see that here the rostrum is changing quite a lot along with the tooth, and here it looks more similar. So again, relating back to our species, we tend to think of subgenus Thymomes as again being more ancestral with relation to body size and its tooth position, with subgenus Megascapus being more derived. And as I'll show in my study, the truth is a little bit more complicated. So again, here's our phylogeny, and we can make several predictions based on body size and the type of soil that gophers are found in. So in that first square, we have these small gophers found in soft soils, which is what we would expect. And then in that other one, we see larger gophers, which, part of the pun, have a head start to dig in those harder soils because they're larger, their teeth are already more percumbent. Second part, however, we have some unexpected conditions here. So this dark blue gopher is small, but is found in those really hard clay soils. And then also we have these yellow gophers over here that are large, but are in those sandier soils that are easy enough to break apart with the forelimbs, so we don't expect a huge selection pressure for tooth digging there. variation, the huge variation that we see in this genus, is due to a combination of functional adaptation to local soil and phylogenetic history in terms of body size. So in order to study this, I used a method called geometric morphometrics, which you heard a little bit about in Kate's talk before mine. But basically it's using a network of these points along the skull to measure shape. And I did this for both the ventral and the lateral view. Today I'll just be talking about the lateral. I was able to use all mature female specimens, so there's no confounding variables with age or sex. And for each of my 10 distinct species, subspecies in my group, I was able to get 25 per taxa, which means that I've created this enormous data set of over 400 skulls. This is about 400 to 500% larger than other studies that have been done in the system, and has allowed me to get really fine-grained differences between this group. So it's one of the most complete studies on this taxa. So in order to analyze shape variation, I used principal component analysis. Each one of those points represents a different individual, and the axes, the analysis pulls out what explains the most variation. And it generates these really helpful work plots that show us how shape is changing along this axis. So I've shown the work plots for the x-axis here. So I'll be walking you through this chart for the rest of my talk. There are 4 stories to tell. I only have time for 3, but ask me about the 4th one in the questions. First is that we're seeing convergent evolution across these 2 subgenera in hard soils. The small-bodied gophers in those hard soils, we're seeing it compensate for its small-bodied constraint. And the third is an example of we're seeing high procumbency in these large gophers, but it seems to be a signal of phylogenetic history as opposed to adaptation. And finally, the last story is about phenotypic plasticity, but I won't get into that today. So the first result about convergent evolution, you'll see again, we have representatives from both subgenera occurring in these really hard clayey soils. And if we look at this PCA, we see that both of these groups are trending away from the rest of the genus towards the right part of the graph on the axis. And if I blow up the chart and bring your attention to the work plots there, you'll see that it has to do with procumbency. And I would argue it's procumbency that's related to a derived tooth position. So I'm going to label this x-axis procumbency due to derived tooth position. And this is a really exciting result for me because as far as I know, and I've been working with these gophers for years, this is the first time anybody has found evidence of tooth digging in the small-bodied genus, or subgenus here. And you see that it's trending with these large-bodied gophers that are found in clay soil. So it's a really strong signal of functional adaptation for hard soil. And again, if we focus on those small gophers, we're seeing that it seems to be they're compensating for their small size. They're falling out further on this axis than the larger gophers here, which are found in harder soils relative to those small ones. And that this, again, is a signal of functional adaptation because phylogenetically, those dark blue gophers are more related to the light blue gophers over here. This is in contrast to the largest gophers that are in this group, these yellow gophers here, which I'll remind you their place in the phylogeny is again with this larger subgenus here. They're found in sandier soils than we expect, so the kind of soils that you don't necessarily need teeth to dig in. So going again back to the plot, I'm going to bring your attention to the y-axis now and the work plot that this generates. Again, it's related to procumbency, but here I would argue that this is procumbency due to size. And we can see that because the rostrum is really changing along with the angle of procumbency. So this is an example of phylogenetic history driving a trait, but since we're not seeing it get any more derived for digging in harder soils, it's not a functional adaptation. So to summarize, we have this really neat story, and I can walk you through this entire graph talking about adaptation phylogeny. First, we have this ancestral group, small body size, found in soft soil, not many changes. So relative to this ancestral condition, we're seeing large gophers that already have high procumbency getting more derived, getting more adapted to those harder soils. We're seeing smaller gophers overcoming a constraint of small size to dig in harder soils. And third, we're seeing an example of phylogenetic history as opposed to adaptation in these large gophers. So it seems our hypothesis stands. I'm happy about that. And with that, I'd like to thank my mentors, collaborators, and research funding. if I could make a shameless plug, I'm also an educational game designer. Some of you have played my game Go Extinct. As part of my Fulbright Scholarship, I'm generating another game featuring Australian mammals to teach kids about evolutionary development. And if you know anything about embryos in Australian mammals, I would love to talk to you about the accuracy of the illustrations. So, thank you. Yes, so that's a great question about lower incisors. In fact, gophers actually use their lower incisors more than their upper incisors, but because of the placement of the cranium and the jaw, the upper have to get recumbent to allow the lower to be fused. So we can infer from the upper incisors that recumbence will be lower.
There's Australian riders with forward facing upper incisors. Are they likely to be dealing with their teeth?
So are they highly recumbent?
Forward facing, so big thing.
Okay.
They live in heavy soils.
Possibly, yeah. So all rodents have been shown to use their teeth to dig at some point. It's especially when they're coming across like vegetation. The primitive condition, however, they have to like kind of crane their neck in order to get perches. So it's really looking